Lecture 5 on the nervous system. Nerves tingle and tell us of what we are not, but what are we but nerves? All right. So the brain, so many of these words that I use, that anatomists and physiologists use, have their root in Latin or sometimes Greek. But the word brain itself is, the word itself is even older than that. It goes all the way back to a Proto-Indo-European word called Meregno. Meregno. I don't know. How do you say that? Meregno. Meregno. No. I don't know. Meregno. I mean skull or brain. And which then became the Proto-Germanic word bragnon, or what we understand to be brain now. So all of that hints at the how early on humans recognized the importance of this organ that resides in our brain case, and how much it distinguishes us from other animals. If you think about the sheer volume of brain to body mass, no other animal comes close to what humans have carrying around. So all this energy that's invested in creating such a large organ in comparison to our overall body mass must pay off somehow. I'm still trying to figure out how. Aristotle, in the 4th century, I've already kind of pointed out his foibles. It's sort of fun to make fun of Aristotle sometimes. He's so adulated. He thought that nerves were controlled by and originated in the heart. And he was, in fact, wrong. These were ligaments. And it was Galen, I think, perhaps I've already said this slide, that maybe that first slideshow I should have redacted that slide, but and saved it for here. But he said he disparaged Aristotle as one who knows nothing of what is to be seen in dissection, and established the fact that it was the brain that was the source of the nerves and our sensation and our voluntary motion. And this is the important term here, voluntary. So there's something about the brain that is associated with our will, our choice, the sense of ourself, our conscious awareness, right? And he calls it the encephalon. So that's the brain. The word nerve, however, does come from Greek. The word neuron itself is meant to mean sinew or tendon. And this is an irony, and again hints at Aristotle's misunderstanding of what nerves were and how much they look like tendons. I think during the muscle chapter I pointed out the freshman nerve, did I? The plantaris nerve. So when the few of you who may get a chance to be in a cadaver lab, you will see how much nerves and thin tendons can resemble one another. Moses Maimonides says, so I had mentioned him previously, the Jewish scholar. One who is not knowledgeable in anatomy may mistake ligaments, tendons, and cords for the nerves. It wasn't until much later, after Aristotle, that his errors were revealed. So Alessandro Benedetti, 5 years after Columbus sailed the ocean blue, said, by means of nerves, the pathways of the senses are distributed like the roots and fibers of a tree. So what we have here is an understanding of the overall architecture of the nervous system. So there is this, the brain itself is sort of the tree, and then all of the peripheral nerves are its roots that fragment and arborize and distribute themselves out into the body. We have Da Vinci, the consummate artist and the consummate Renaissance man, said, "The medulla is the source of the nerves which give voluntary motion to the members." All right, so he thought that the medulla oblongata, the oblong medullated part of the brain, he believed that was actually the source of our volitional control over our muscles. It turns out he was wrong. They were just passing through that. But it was beginning to be understood. This is the beginning of understanding the correlation between neuroanatomy and the function of the central nervous system. So what is that function? Well, it is first to derive information about the external environment. All right? If we're going to be conscious, what does it even mean to be conscious? Consciousness importantly involves an internal representation in some manner of an external state. All right? There's something external to an organ. There's a boundary, and there is an external state, and we're going to internally represent that external state using whatever means necessary. And this is the function of our sensory organs, to collect information about the world around us that we are coming in contact with. And then once we've gathered that information, we are going to internally represent it and integrate that information. This is the job of the brain and to a lesser extent the spinal cord. And then once that information has been represented and processed, integrated, there's going to be an output that gives us some sort of control over what are called effectors. And an effector can be a muscle, it can be a gland. There are different types of effectors, and we'll see them in a bit. So what are some of the parts of this? There's going to be peripheral nerves, which connect to a spinal cord, and they may pass through ganglia. Ganglia exist in the peripheral nervous system. What did I say ganglia were in lecture today? Yeah, bundles of nerve cells, the soma, the cell bodies of a bunch of peripheral nerves. In ganglia, pass through ganglia into the spinal cord and then ascend the central nervous system to the brain, or not. So here is a kind of a hierarchy of the nervous system. So we have the central nervous system that has the brain and spinal cord. The peripheral nervous system, and these are going to be the nerves that radiate out from or converge upon the central nervous system. There's going to be two broad categories, either somatic nerves or autonomic nerves. And somatic nerves are going to be ones, the kind of nerves that control muscles, voluntary muscles, for example. And then the autonomics are going to be, and this is going to be tend to be volitional. The autonomic nervous system is going to be part of the brain's information gathering and control that is subconscious, meaning that you don't have volitional control over it. Subconscious meaning you may be aware of you're going to be aware of the activity of the autonomic nervous system. You're not unconscious of it, but it is below your volitional control. This is broken up into the sympathetic and parasympathetic nervous systems, the fight or flight versus the rest and digest nervous systems. And then lastly, I'm not going to get very much of an opportunity to talk about it, but there is the enteric nervous system, which is still connected to the central nervous system, but fairly independent. Enteric meaning related to the digestive system. All right, so this is going to control a lot of digestive processes that happen in the stomach and the intestines and whatever. And it's a little bit of a parallel portion to the nervous system. So in terms of the nerves that are in the peripheral nervous system, you can have, there's these three different dichotomous distinctions. There's these three different dichotomous options that you can have. So you can either have general nerves or special nerves. Okay, what does that even mean? We'll talk about what that means a little bit. But it's one distinction. So there are very general nerves that are generalizable to broad swaths of the body, to relatively divergent effectors with divergent functions. Or you can have very specialized nerves that are only going to serve one very narrow function. They serve a single narrow function. Somatic versus visceral. And this is kind of the distinction between somatic and autonomic to a large part. All right. So somatic tends to mean that it's going to be serving your skeletal muscles or the like tactile senses. All right. And then the third dichotomous distinction is afferent or efferent. Afferent meaning sensory, efferent meaning motor. So afferent ascends, efferent descends from the central nervous system. So that means since there are 6 options, there are going to be 8 combinations of each of those 3 dichotomies. Just like a cube, 6 sides, 8 corners. All right. So we can have, for example, general somatic afferents. A general somatic afferent nerve would be like Meissner's corpuscle in your hand that gives you the sensation of touch. Or special somatic afferent or special visceral efferent. You know, there's all kinds of, there's 8 different types of nerve fibers. I'm not going to go through each of them in detail, but special somatic efferent may or may not actually exist. It's a part of contention, a point of contention amongst anatomists. Some people say that there are distinct SSE fibers, and some people say no, they're all GSE. But that's way beyond what I'm going to talk about. All right. So I've already, this is kind of the same thing I've been saying the last few slides, but I'm going to expand it a little bit. So here are the sensory neurons. This is the information gathering neurons that are they're afferent sensory neurons, meaning they're ascending. So it's the difference between affect and effect. If you are affected by something, something happens around you that's going to cause a change in you. It's something that's coming up into you, right? Affected versus you cause an effect. You bring about an effect. You change something that's outward going, right? You cause an effect. So affect, you are affected and you cause an effect. So an afferent neuron is a sensory neuron that is bringing information up to your central nervous system. There are 3 types of afferent neurons: interoceptors, exteroceptors, and proprioceptors. First category that I have listed here is simply interoceptors. And interoceptors are going to be sensory receptors that are going to return information about your internal state. All right, so your stomach and the viscera, the visceral compartment, compartments. Oftentimes, interoceptors have referred sensitivities. So you can have like referred pain that is being triggered by interoceptors. Exteroceptors. And there's also internal senses like taste, deep pressure, and pain are considered to be interoceptors. Exteroceptors are simply the external senses, so the things that are the closest to the surface of your body. So light touch, temperature, light and pressure. Any of your distance senses, such as sight, smell, hearing, things that give you a sensation at a distance, external sensory input. And then last is proprioceptors. It's qualitatively different than the others. Proprioception is, for example, if you go, well, we all went to the museum. Did you guys see that totally amazing picture of Michael Jordan doing that slam dunk? Huh? I took a picture of that, showed it to my son. He was watching me try to make the art and anatomy thing for you guys, and I was considering that picture. He's like, he's flying through the air. That guy has a pretty remarkable proprioceptive sense. It is a sensation of the position of your joints, the position of your limbs, your body, like what is the tension in your muscles, what are the specific position of all of your joints. It's related, it returns the kind of information that's important for balance and coordination. So proprioceptors. So that's the sensory information. Then there is the interneurons. Interneurons, it turns out, are the bulk of the neurons in the brain, or in your body. Bulk of the neurons in your brain are interneurons. And it's what I'm going to talk about the very least with you guys. Because it's pretty wide ground. You can take a neurophysiology class, a neuroscience class, and learn more about interneurons. But they do a wide panoply of functions that we associate with, our human existence. So everything from memory and learning and imagination and speech and whatever, the integration of all kinds of information. What makes us are the interneurons. And then finally are the motor neurons. These are the efferent neurons that are going to cause an effect with their action on an effector being some sort of cell that's going to do something. Maybe it's a glandular epithelium that needs to get turned on, or maybe it's a muscle cell or whatever. The two types are somatics. These are the skeletal muscles by and large. And then the visceral effectors. These are synapses that happen at different autonomic ganglia. So they can be either sympathetic or parasympathetic ganglia. Because of that, because there's this synapse at a ganglia in a visceral effector, there's a ganglia, there's going to be a preganglionic fiber as part of the peripheral nervous system, a preganglionic fiber, so a fiber connecting the central nervous system and the ganglia, and then a postganglionic fiber that just got synapsed on by the preganglionic fiber, and that's going to directly connect the effector. All right? So there's one little extra step in these visceral effectors. It's this synapse. All right. So the divisions, continuing the same story. We have these afferent neurons or sensory receptors. They can either be going directly into the brain via what are called cranial nerves. There are 12 cranial nerves. I probably won't get a chance to cover them today. And then there are peripheral nerves that enter directly into the spinal cord via these spinal nerves. So there's two ways into the central nervous system, through the spinal cord or directly into the brain. If you're going directly into the brain, you're one of only 12 nerves. And then there's this host of different spinal nerves that go in along each of the spinal segments. You can have reflex centers in the spinal cord. You can have reflex centers in the brain. If I'm lucky I'll be able to talk about reflexes at the very end of class today. And then you have motor output that can either come along cranial nerves or along the spinal nerves. So for example, here, taste is purely along a cranial nerve. You have the facial nerve that brings taste from the anterior 2/3 of the tongue up to the brain along the facial nerve, cranial nerve 7. And then there's a processing center in the gustatory cortex on the temporal lobe that says, oh, that tastes delicious. Time to make some saliva. And then it sends motor output along the facial nerve to the submandibular and sublingual glands that produces saliva in the front of your mouth. Okay? So these motor nerves come to various effectors. The one I just described would be some kind of a gland, the submandibular gland that's going to produce some saliva. The spinal cord is going to do the same thing. You're going to have sensory receptors, touch, whatever, that comes into the spinal cord. Perhaps there's a tack. You step on the tack, and a spinal nerve sends a signal to the spinal cord. There's a reflex center. Hopefully that's happening faster than I can talk about it here. And it sends motor output to some muscles that make me jump off the tack. All right? Does that make sense? Very straightforward. Okay. So I'm going to go through that cycle that I just described step by step and describe each of them in more detail. Sensory receptors first. So sensory receptors are going to send 4 types of information. Every sensory receptor is going to give you 4 types of information. The first is modality. Is it taste? Is it touch of some sort? Is it hearing? Is it smell? Is it sight? Right? These are the 5 senses. And in fact, there are those are main general categories. There are subcategories within those that, so there's actually a lot of different types of sensation. For example, this is probably the broadest category, touch, right? We have temperature sensitivity. We can tell something's soft. There's light touch. There's deep pressure. There's vibrational sensitivity that you can have. There's a lot of different tactile modalities. So modality is one kind of information. How does your brain keep it straight? Well, there is this thing called labeled line code. And for example, a lot of people, there was for a long time a myth that the tip of your tongue is sweet and the sides of your tongue are salty or whatever it was. People had these different maps for taste on the tongue. And that's actually not the case. That's not how it works. Every taste bud is going to be able to taste the 5 categories of sensory receptor. These are bitter, salty, sweet, umami, and sour. Bitter being things that are alkaloids or basic in pH. So for example, caffeine is something that is bitter. And salty, I think we're all familiar with sodium chloride. Something that's sweet, I think we're also familiar with. Umami is the sense of savoriness. We call it umami because the Japanese were the first to identify those sensory receptors. And in fact, Japanese cuisine had already had a word for it. They called it umami because Japanese cuisine has a lot of this flavor in their food. And these are sensory receptors that are triggered by glutamate. Glutamate being one of the amino acids. So and glutamate is a proxy for protein content. So if something has protein in it, specifically glutamate, it's going to be tasty and set off the umami sensation. And then sour is the opposite of bitter. Sour is something that is acidic, like a lemon would be sour. All right. And any taste bud can be triggered by any of them. Say these green neurons here represent sour. We eat something, we lick a lemon, and the neuron that the signal is coming through to go up to the brain, that line, the brain has learned to interpret as sour. It has learned to interpret that as sour. So So has anybody heard of the miracle berry? Yeah, it's cool. You ever had one of those?
No, but my friend just got some, so I'm gonna do it soon.
Yeah, you should. It's fun. I do it with my biochem food class. It's pretty cool. There are these berries that they mess with your sweet receptors in your mouth, and they mess with it in such a way that acids, things that are sour, trigger the sweet receptor. All right, so even though you are eating something that's sour, it's being interpreted as sweet because it's this line that is being triggered. So, for example, lemons taste delicious, like lemonade, and strawberries are kind of repulsively sweet. The second type of sensory information that is being transmitted is, of course, location. All right. So location implies something about the brain. It implies that there must be a somatosensory map. Your brain must have a map of the body somewhere represented in the brain that this sensory information is lighting up. So I touch somebody in the shoulder, I give you a pat on the back, and it's lighting up sensory neurons that are associated with the back. All right, so a neuron is transmitting information about location. Here is this relationship I have. The size of the receptive field, so there's one neuron that's going to have some receptive field, is inversely proportional to your ability to tell where it is. Okay, so for example, you have very small receptive fields on your fingers, places like your fingers, your lips, your external genitalia, whatever. This means that the surface area of skin has a high density of neurons. All right, a high density of neurons. So if you poke the skin, oh man, it's tomorrow. That's part of the lab's tomorrow. Sorry, we're going to do this experiment in the lab. I forgot what day it was. It's tomorrow. Where you poke the skin on your finger with these calipers, and you can tell that it's two different things poking you. Whereas you do it on the skin on your back, which has very large receptive fields, and even though they are spread far apart, you can't tell that you're getting poked by more than one needle. In fact, you can map the size of the receptive field with this tool that I have up in the lab. We can map the size of the various receptive fields on places on your body. Kind of fun. Third bit of information that a neuron's giving you is the intensity of the stimulus. So there's a couple different ways that this can happen. But all of this intensity information has got to be frequency encoded, right? It's all frequency encoded because these are action potentials. I talked about amplitude modulation and frequency modulation earlier. So this is all frequency information. So, and there's three different ways. So what fiber is it that is sending that signal? Maybe this neuron here that I show, the specifics of its membrane dynamics means that it's got to take a really intense stimulus to get it to send an action potential. All right? So that's not going to send as much. It's going to be a relatively insensitive neuron. All right? Next, how many fibers are firing? Are we going to send more than one fiber? Those action potentials are going to be additive in their effects on the brain. And then how fast? So some neurons themselves are going to, depending upon the stimulus that they're gathering, they're going to be firing more quickly or more slowly. All right? And so that's another way that intensity can be encoded as it is sent to the brain. So we've got modality, how does the brain interpret it? Location, how is it mapped onto the body? And then the intensity, how intense is the stimulus that the neuron is gathering. And lastly is duration. Duration. So to talk about this, I have to introduce a concept called adaptation. So adaptation is the brain's ability to reduce your sensitivity to stimulus to a constant stimulus. You know, if you were to ask a lot of people who perhaps haven't thought very deeply about the brain, what the main job of the brain is, you might imagine, or the sensory portion of your brain, you might imagine that people would say the job is to gather as much information about the external world as you possibly can. That's actually not the case. The brain is trying to filter out all of the chaff. It's trying to find signal in the noise. To find signal in the noise, you're trying to turn the noise down. Stimulus that is not meaningful is filtered out. One of the brain's greatest achievement is not to collect a huge amount of information. It's to filter out what's meaningful and what's not meaningful. All right? So for example, as we sit here, let's take a moment of mindful silence and just be in the room together for a bit. All right. What do you hear? I hear the air handler. It's been there every day that we've been in lecture, for every minute that we've been here. Have you ever paid attention to it before? Likely not. Yeah, very likely not. Because your brain has adapted to that sound and filtered it out. You are able to focus on my voice as being meaningful noise, hopefully, and the air handler as being meaningless. Okay, so that's adaptation. The sound of the fan became meaningful. Yeah.
But then what kind of agency does that leave us? I mean, if we can't choose what we filter out, then how much control do we have over ourselves?
Mm-hmm. Yeah, it's. I asked you to be mindfully silent, and then, and we were within the context of this conversation, it became obvious to you that there were sounds, and you heard them, and I drew your attention to it. So you had the agency to choose to pay attention to that sound, right? You had the agency to choose to pay attention to that sound. However, we are biological creatures, and we do not have, there's no point in having our volition burdened by making decisions over things that can be automated, right? Like, if you had to choose every moment to not listen to that fan, you would lose your mind. You would lose your mind, right? So there are things that are best left to automation. Yeah, adaptation.
Does it go the opposite way too? Like, can you choose what to adapt to and what not to adapt to? Like, can you choose to not adapt to something or to adapt to something?
Okay, well, certainly there is biofeedback, and that opens up the door to a much larger conversation. But there are specific neurons whose very structure, they're like the kinds and distribution of proteins that they are expressing, the receptors on the surface of that neuron are going to be geared themselves towards types of adaptation, all right? And in fact, the nervous system itself, God, like, I really would love to teach like a 300 level class about this, so I don't have to feel like I'm just trying to get through the basic information, because this is so fascinating. It's such a fascinating topic. But so, for example, you can take, if you think about you can describe the pattern of any kind of stimulus mathematically by looking at something called frequency density, and it's just looking at a normalized frequency range of a signal. You can describe those as pink noise or white noise or brown noise, different types of noise. White noise is purely random, and pink noise is kind of random, a little bit predictable. Brown noise is more so. All the way on this gradient, all the way to like a pure tone, which is utterly unrandom and pure and utterly predictable, right? So you can take an electrode and put an electrode that's going to put a stimulus on your finger with some kind of frequency density profile, and then you can take another electrode and put it at the brainstem as the signal is entering the volitional part of the brain, and you can compare those signals. And so perhaps you're going to put something that has a brown noise, Brownian motion, a brown noise signal on the finger. If you were to look at the frequency density of the signal that's coming to your brain before it even reaches your awareness, your nervous system has already, by virtue of the synaptic connectivity and the biophysics of the neurons, filtered that to allow the signal coming through to be more pink in its frequency density. Pink noise are patterns that our nervous system deems to be more meaningful types of noise or signal, right? So outside of our volition, our nervous system is just geared. It's just built right into the system as as signal filters. There's different filters. Does that make okay? So that is some nuanced stuff there that I'm not necessarily going to be teaching, expecting you to know, but I do want you to know what tonic and phasic receptors are. Tonic receptors are ones that are simply slow adapting. All right, once they get turned on, they're always on. They show very little peripheral adaptation, whereas a phasic receptor is one that is rapidly adapting. So this tends to be in situations where we need information about the change of rate in stimulus. So basically, you're looking for first derivative optimizations for people who want to think about it like that. This is things like smell, hair movement, cutaneous pressure. As you're sitting there in your chair, like I'm going to sit on the table here. I feel the counter on my butt, but if I'm going to just sit here for a while, I will rapidly forget that I'm sitting here, right? That I feel the chair. Or if I move my hand through my hair, I feel it as I'm moving my hand through my hair, but then I don't really feel it quite the same way any longer, right? Rapidly adapting. Phasic. Whereas tonic receptors, these can be things like body position. So when I move my arm over here, like as I'm talking to you, I know my arm is in this position the whole time. I don't forget that my arm is sticking out over there. All right? If I were to close my eyes, I wouldn't be like, oh my God, where are my arms? Where are my legs? I can't feel them any longer. Hopefully, shouldn't. They're tonic. All right, so if you look at the receptor potentials, so here would come a tonic signal, and it would stay depolarized. The nerve, not the neuron, the nerve would be continuing to fire until you release the stimulus, and it would drop back down. Whereas a phasic, when you get a spike, it would come back down to rest, and if you release the pressure, you'd have this off response. All right, so that is duration. Those are the 4 different types of information that are transmitted. Here are some classifications of the afferent neuronal categories. So this is all just sensory nerves. We have the fiber types. I told you there were the 8 fiber types. Those are the 4 that are sensory. That is the A, the 3rd letter in those 3-letter designations. The distribution is intero, extero versus proprioceptor. All right. So there's fiber type. There's the distribution. Then there's modality. what kind of sensation is it being interpreted as by the brain? Is it temperature? So these are the general versus the special senses. Is it temperature, pain, touch, pressure, vibration, or proprioception? Those are all general sensations. That's the G in those four. Or the special senses, olfaction, sight, taste, equilibrium or balance, which is an inner ear thing, and then hearing. That is the first S in the blue box up there that corresponds with this blue box. All right? And then these are the different functional categories that we have. The word nociception, N-O-C-I-C-E-P-T-O-R, nociceptor, is a pain receptor. Pain is an interesting sensation because it turns out that, and this is a good evolutionary development, it turns out that all of the sensations that you can experience are going to have some sort of mapping in the brain, right? There's going to be some neurons in the brain that if you light it up, you're going to smell almonds. Or if you like you take a probe and you stick it in there, your arm's going to twitch or something like that. But nociception, there's not a place that you can stick an electrode in the brain and you're like, ow, that hurts. It's a complex sensation that, and I'll leave it at that. There are two types of fibers, type A and type C, two types of nociceptive fibers, fast and slow. Fast and slow. Second type of, second functional category are thermoreceptors. These are temperature receptors. Chemoreceptors can be responsive to the presence of different chemicals. So taste would be an example of that, or for example, things that monitor carbon dioxide levels in the blood in your aortic arch or the carotid bulb. Meccano Receptors, I talked about those a little bit on Wednesday of last week, where I was talking about the deformation of the membrane, opening up a receptor. This breaks down into three types. There are the tactile receptors, which are touch receptors. The baroreceptors is a kind of mechanoreceptor. This is monitoring pressure, like blood pressure, for example. And then proprioceptors, we've already talked about. Those are the ones that are in the joints and the muscles that report on body position. Okay, so there's like basically the outline of everything. I'm going to try to go through as much of it as I can without going through too much. All right, I'm going to point out a few of the important ones. Not all. I can't get all of it. But so here are some general. Another word for general is somasthetic. Somasthetic. S-O-M-A-S-T-H-E-T-I-C. Somasthetic senses. The general or somasthetic senses. There's three types of mechanoreceptors, as I had on the that black box on the previous slide. The first of them that I listed were tactile receptors, and there's two categories of these. First category are free nerve endings. Here are some examples of that. We can have totally free nerve endings. These have extremely small receptive fields. So pain and temperature are the typical sensations sent by the free nerve endings. Why does it make sense to have small receptive fields? What would the problem be with having a large pain receptive field? A large receptive field for pain. Yeah, you don't know exactly where it is. Maybe something's biting me. Where is it? You want a small receptive field. Similar with touch. There's a problem. If you're lighting off a nociceptor or a temperature receptor, this is typically not a nice thing. Something may be wrong. Yeah, Liam, you have a question? Deep thought. Okay, nope. Next is the root hair plexus. And the root hair plexus is kind of similar, except it's in the way the brain interprets it. It's this nerve ending that wraps around the bulb of a hair. All right? So when you run your hand through your hair, when some critter's crawling on your your arm hair, you're triggering these nerves. Next is the Merkel cell. And so I'm going on this gradient of more and more specializations to the nerve, accessory structures that are going to modify the sensory information we're gathering. So we have Merkel cells. Merkel cells are ones where the sensory end, the business end of the dendrite, is associated with these what are called Merkel cells. So here's a tactile disc that is actually part of the neuron, and the Merkel cell is a specialized epithelial cell that is going to trigger the tactile disc. So very, very light. The lightest touch that you can perceive is that information is transmitted by Merkel cells. All right, very, very light touch. Maybe a bug is walking on you, or someone you like is touching you softly. All right, so the next category are nerve endings that are encapsulated. All right, they're surrounded by some sort of thing that's going to attenuate the responsiveness of that sensory neuron, meaning it's going to reduce the sensitivity. So we have a tactile or Meissner's corpuscle. Here's one. Here's the nerve ending, and it's surrounded by this collagenous capsule, this collagen capsule. They're pretty large. They're fun. I often put them on the lab tests because you can see them really easily. You can identify them very easily in tissue sections. And they embed in the papillary layer of the dermis right beneath the epidermis in the ridges. And it gives you, again, light touch information. Also low frequency vibration and pressure. These are relatively rapidly adapting neurons. It takes about a second for them. So does that mean they are tonic or phasic? Phasic. Good. Thank you. Pretty big things. So these are the ones that you're going to find in your eyelids, your lips, your fingertips, nipples, external genitalia, all the sensitive parts of your body. They tend to have very small receptive fields as well, and consequently have large real estate devoted to them in the brain. Next is the lamellated, or eponymously Pacinian Corpuscle. Pacinian? I've always heard Pacinian. Pacinian Corpuscle. This is a single nerve or fiber that looks like it's wrapped up in an onion. There's like all these layers. And the Pacinian Corpuscle sits in the dermis, in the deep dermis at the hypodermal boundary. This is going to give you the sensations of deep pressure. Deep pressure. So any kind of pulsing or high-frequency vibration as well. These are also phasic receptors. Deep pressure. Next category, class of mechanoreceptor or baroreceptors. I'm not going to say much about them. Proprioceptors are the third. We have three types: muscle spindle apparatus. So this is something that's embedded actually in a muscle. There's the Golgi tendon organs that are in the connective tissue of a tendon. And then there are receptors that you find directly in the joint capsules. So different types of proprioceptors. Interesting topic that I'm not going to do anything but mention. I won't go into any of that. Oh, I will say this. If I get to it, which I'm doubting more and more as the lecture goes on, these muscle spindles are what are responsible for the muscle stretch reflex. If I get there, it's the muscle spindles that are responsible for that. Chemoreceptors. So they live mostly in the aortic bodies and the carotid bodies. So the carotid bulb is right here in your neck, and the aortic bodies are in the aortic arch. And they are going to monitor a number of metrics like pH, carbon dioxide, oxygen levels in your arterial blood. Is it possible? My hands are cold. I don't know. Cranked it up a little bit. I'm not sure if that's going to do anything or not. Got a bunch of like hamsters on the wheels over there in the physical plant running faster. Okay, carbon dioxide, pH, and oxygen. So what can happen here is these neurons can adapt. Their peripheral adaptation can happen over a period of seconds, but we can get central adaptation can also happen. So I didn't get a chance to talk about Lou Gehrig's disease and my dad, but one of the things when I've had a number of friends have Lou Gehrig's disease, and it's interesting to watch them as they have ventilatory insufficiency. They can't breathe, they can't move oxygen very well, so they become hypercapnic. Hypercapnic. They have too much CO2 in their blood, right? There's too much CO2 in their blood. It makes them a little loopy towards the end. And their brains adapt to this higher level of CO2. These receptors are saying, hey, there's a problem here. But the brain just says, okay, I can't, whatever. I know, I've heard about it. It's been a continuing problem. So there's central adaptation that can happen in the integration center for the homeostasis, that homeostatic system that controls response to elevated CO2. All right, here is what I think it was Alan or somebody was asking about. No, it was Annabelle, I think. Is that right? I don't remember. Somebody was asking about the different shapes of the neurons. Yeah, and you were so both of you, the two of you, and you were asking how can you tell the difference, blah, blah, blah. So here are the structural classifications of them. We have, the first is anaxonic, A-N, meaning no axon. The anaxonic neurons have no actual axon. Well, that's not true. There is, so most of this is input. One of these is output, but morphologically, you cannot tell the difference. Yes, sir?
These are astrocytes?
Nope. Astrocytes are glial cells. They look like astrocytes. Yeah, glial. We'll get to those in a minute. Then there are bipolar neurons. Bipolar neurons have one input and one output. I feel like Christopher Robin with my little stick sword up here. Unipolar neurons look similar to bipolar neurons, don't they? But they're not. They're not the same. You'll notice that in a bipolar neuron, you have information coming in from one source. Information coming in from one source, passing through the cell, passing through the perikaryon, impacting that electric field that's passing from the dendritic input is impacting in whatever way it does the structures in the cell body. And then there's whatever processing event happens, then there's a direct transfer out. So these bipolar neurons do have some sort of processing event that happens. Whereas unipolar What are these? These are basically just cable extensions, aren't they? Signal comes in, doesn't pass through the cell body, and just keeps on going through the axon. Unipole. The perikaryon does not play a role in these cells. And then lastly are the multipolar neurons that we discussed in lab today. All right, so those are neurons. But actually, half of the volume of your nervous system are not neurons. They are not cells that whose job it is to retrieve information and send signals. We call them neuroglia or glial, G-L-I-A-L cells, glial cells. So they're both in the peripheral and the central nervous system. First, I'm going to go through the central nervous system. There's 4 types of neuroglial cells. First are the ependymal cells. Ependymal, E-P-E-N-D-Y-M-A-L, ependymal cells. The ependymal cells line the ventricles of the brain and the central canal of the spinal cord. Their job is quality control, to monitor the characteristics of the cerebrospinal fluid that is circulating throughout those void spaces. Next are the astrocytes. Astrocytes. Along with the oligodendrocytes, the astrocytes help to maintain this blood-brain barrier that I mentioned earlier. More than that, they also provide structural support. So the thing about the brain is it's not just a big gamish of brain cells. There is a spatial structural hierarchy that is closely maintained, and it's the job of the astrocytes. Thank you. Pardon me. To maintain that three-dimensional structure. So because they're maintaining the blood-brain barrier, they are going to be in control of ion flux, of whatever kind of nutrients are getting in. They're the gatekeepers. They're kind of the gatekeepers that are going to determine what is going to reach the brain and not reach the brain. This is what will allow some glucose to get through, or not going to allow these particular chemicals that we don't want near whatever hormone is going around the body. Something that you don't want near the brain is not going to get past the barrier provided by the astrocytes. The job of the astrocytes is also to absorb any stray neurotransmitters that may have been released by neurons and to recycle those. They also form scar tissue if you have had any kind of brain injury, infarction, something like that. All right, oligodendrocytes. We talked about Schwann cells in lab today. Gotta keep moving. The oligodendrocytes, they are the myelin in the central nervous system. So what's different between oligodendrocytes and Schwann cells is that oligodendrocyte, if you break the word down, oligo means several, dendro meaning projections. Dendrites are like the branches on a tree, and cyte, cell, so these many branched cells. Oligodendrocytes, so a Schwann cell wraps around and does its business in one interneuron on an axon, just in one spot. But an oligodendrocyte is a cell that has many arms that reach out, and each of those arms forms a myelin sheath on different axons that are all nearby to one another. Why do you think the central nervous system is like that? What do you guys think? Yeah.
It secures their positions.
Yeah, absolutely. It again helps maintain this three-dimensional hierarchy, this three-dimensional structural aspect to the nervous system, which I indicated was so important. And then lastly are the garbage trucks. Everybody needs somebody to come up and clean a mess up for them sometimes. So the microglia are simply these white blood cells that these little specialized cells for the nervous system, whose job it is to go around and clean up any kind of cellular debris. And if there are pathogens, to hopefully deal with them. There shouldn't be, but all right. So here's a picture of some gray matter. Gray matter because here's the soma. We have some oligodendrocytes that are on the axons. We have these astrocytes here, which are covering up. They're covering up the surface, creating a blood-brain barrier. Here's this. This is a capillary. It's entirely encasing that capillary. Ependymal cells, which are in communication with the void space of the central canal or a ventricle. Little microglia in there looking for something to do. And then if we were to look at white tissue again, here's that capillary with the astrocytes covering it and controlling what gets in and out. And then, so when I say this is not to say that there isn't, it's not like the brain itself is covered in a blood-brain barrier and there's no blood inside the brain. That's not how it works. There are blood vessels richly filling every corner of your brain, but those blood vessels have an extra layer that regulates what can get out of the blood vessels, or extravasate is the word. But what is an exchange in this capillary and the extracellular fluid that the cells themselves are exposed to? All right. Yeah. Picture of an oligodendrocyte wrapping itself around the cell. So here is the endoneurium. Here's this neurolemma. This would be axolemma right here. But the neurolemma is this outer membrane of the myelin sheath, and then the endoneurium is the connective tissue sheath on it. You can see what a beautiful picture that is at the top. All those different layers of myelin wrapping around. Right. So same story in the peripheral nervous system. We have neuroglia there, except we have only two. And these are the Schwann cells, which I've already talked about, which just wrap themselves around the axon, and these satellite cells. The job of the satellite cell is similar to the astrocyte in that they're going to regulate oxygen and carbon dioxide levels, nutrients, neurotransmitter, etc. Whatever you're going to find accessing the cell bodies in various ganglia. All right. So we went through all of that. So I went through the anatomy. Should I spend a couple minutes talking about the function of the different parts of the brain a bit? I think so. Well, I went through the cerebrum already. Basically consciousness and subconscious regulation, memory, etc. The diencephalon is important. The thalamus we'll talk about quite a bit. So for sensory, and like write this one, inscribe this one on your brains right now. Hopefully this is going through your own thalamuses. That for something to reach your conscious brain, there has to be a synapse in the thalamus. Anything. For you to be aware of it, it has to synapse in the thalamus. The thalamus is not sufficient for consciousness, but it is necessary. Does that make sense? So for you to be aware of something, some kind of sensory input, it has to synapse in the thalamus. All right, and then it projects out of the thalamus to higher cortical structures where you actually become consciously aware of them. The hypothalamus, right below it, is a complicated region of the brain that does a number of things. It controls the pituitary gland. It's involved in generating emotions with other regions of the brain, regulating emotions. There are various autonomic functions that are controlled by the hypothalamus. The mesencephalon, the midbrain. So in my little list of things here, it says processing of visual and auditory data. Actually, that's through the colliculi, the superior and inferior colliculi back there. That's what that is referring to. You need to have, if you have a lesion in the midbrain, in the mesencephalon, you are going to lose consciousness. You will lose consciousness. That should make sense, though, because signals have to go through the midbrain to get up to the thalamus. So the pons and the medulla, have, so as you kind of work your way down the brainstem, you get to more and more fundamental functions of the nervous system, more and more basic functions of the nervous system that turn turn different things on and off in the body. For example, your stimulation of breathing; those signals originate here in the medulla oblongata, or cardioregulatory system, the part that regulates the speed of your heart, or your various digestive functions. All of that, the really fundamental, basic to survival functions. This is why strokes that happen in the brainstem are often quite catastrophic. And then lastly, the cerebellum. I talked about that in class. The volume of the brain is anywhere from 750 cc's to 2100 cc's across lifespan. And 97% of the neurons in the brain or in the body reside in the central nervous system, the brain someplace. Weighs about 3 pounds. All right. So I already showed you the ventricles, so I'm not going to go through the anatomy of them very much, but I am going to talk about cerebrospinal fluid. It circulates around the brain and is produced by the choroid plexus. I told you that in lab. But it's resorbed by the arachnoid granula, the arachnoid granulations. So if you were just producing fluid without resorbing it, it would put greater and greater pressure on the brain. And in fact, does anybody know what that's called? Hydrocephaly. Yeah, hydrocephaly. What does it do? Well, of course, it gives buoyancy to the brain. Buoyancy. You don't want to be banging your brain around. Buoyancy and protection. It's been a remarkable shift in consciousness in professional sports, particularly in football, since I was your age until what it is today, of the importance of not banging your brain around. It does have a real impact on your cognitive ability later in life. It also leads to the chemical stability. It contains nutrients and various hormones, chemical messengers, and then whatever metabolic byproducts are produced by the neurons are contained in the cerebrospinal fluid. So how does it actually work? Here are the meninges. I talked about the 3 meninges in lab. There is the dura mater, the durable mother on the outside. And here is the falx cerebri and the longitudinal fissure between the 2 hemispheres. And below it is the arachnoid mater with these fine hair-like, spidery web, hair-like projections that travel down to the brain. And then bubbling up from it and piercing through the dura mater into the superior sagittal sinus and the other sinuses. It's showing it in this diagram, but it happens into the other venous sinuses, are these arachnoid granulations, these arachnoid granula. And this is where the cerebrospinal fluid that was made by the choroid plexus gets resorbed into the superior, into the venous drainage and taken away from the brain. So people with hydrocephaly, there's a problem with this mechanism. The final layer of connective tissue is called the Pia Mater, or little mother, Pia Mater. And it's just a tissue membrane that lies on the surface of the brain and kind of helps helps be a substrate for the vasculature, the microvasculature of the brain. So here is just a depiction of that circulation. All right, we have here's some arachnoid. Pardon me. Choroid plexus on the surface of the thalamus there, or wherever, and it's producing cerebrospinal fluid, which gets pushed down the cerebral aqueduct out here into the external space around the brain, and is eventually going to get resorbed by these arachnoid granula, as you see depicted right here. The circulation of cerebrospinal fluid. Blood-brain barrier. This is created by astrocytes primarily, but there's also the capillary epithelium. So the inner leaf of it is the capillary epithelium that would be part of any kind of capillary, right? But then surrounding it and controlling what is able to come in and out of the capillary are these astrocytes, and there are ependymal cells that also form a portion of that barrier. Cranial nerves. There's so many other things I want to talk about. I'll point out a couple of them because we will talk about a few of them. The ones that you need to know about, there's the olfactory nerves. These are the olfactory cranial nerve 1. So the cranial nerves are numbered from 1 to 12, moving anterior to posterior across the brain. All right? And how the number is related to how they exit the brain case anterior to posterior. So these are number 1. They sit, the olfactory bulbs sit on the cribriform plates of the ethmoid on either side of the crista galli, send the rootlets down into the nose. Maybe I have it all on one slide here. Why don't I do this? I had hid these other slides because I hadn't intended to talk about them, but I'm going to show the ones that I actually want to talk about. So I'll reveal them all, and you'll forgive me for skipping past a few of them, right? Yeah. So how do I? I don't know. I'm going to probably have to do this again. I need to figure out how to go directly into the proper view. So the olfactory bulbs are cranial nerve 1 that that sit on the cribriform plate right here, sending rootlets down into the nasal cavity, and odorants come in and trigger those. So that's a good thing to know. Here is cranial nerve 2, the optic nerve. We'll need this for our discussion tomorrow. This is what conducts sight back to the optic chiasm. There's several nerves that control the extraocular eye muscles. Let's not worry about those. We're not going to worry about the trigeminal. I talked about the facial a little bit today, didn't I? But don't worry about that. Vestibulocochlear nerve we will talk about tomorrow as well. This controls hearing and balance. The cochlear nerve for hearing and the vestibular nerve for balance. Glossopharyngeal nerve we won't worry about. The vagus nerve we will talk about. Vagus nerve is cranial nerve 10. It comes off the medulla oblongata, and it's the wandering nerve. If you were to see the full distribution of the vagus nerve, you'd be pretty impressed. Here it is. It goes all over the place. It hits the lungs, it hits the heart, it hits the stomach, it hits the pancreas, the abdomen, the mesentery. This is why they call it the vagus nerve. Vagus meaning vagrant, the wanderer, the wandering nerve, the vagus nerve, cranial nerve 10. All right, so let's slide past all that. Here are the spinal nerves now. So we've talked about the cranial nerves, just the ones that are relevant, and just to mention what their names were, and the various spinal nerves. So the spinal nerves are named for the portion of the vertebra that they're associated with. So there are 8 cervical spinal nerves, cervical meaning cervicis, the neck. And when we look at them, we see here's the posterior, the lateral, and the anterior gray horns, the columns. Here's the dorsal and ventral root, the dorsal root ganglion, the spinal nerve coming together, all of the anatomy we talked about. And this forms this plexus. A plexus is a meshwork or a network of nerves that come together, come apart, come together again in this complex meshwork of trading fibers. There's this cervical plexus that does a wide array of things servicing the head and neck region. Importantly, one of the most important things about it is this thing called the phrenic nerve. The phrenic nerve innervates the diaphragm, which initiates breathing. All right, comes off of C3, 4, and 5, which means if you break your neck above C3, you are in trouble. You are in trouble. You better get that person on a ventilator quick, or they're going to die. Right? If you break below like C4 or 5, you're going to be quadriplegic, meaning you're not going to be able to move your arms and legs, but at least you can breathe. So you don't want a high neck break, because you will lose the ability to breathe because of the disruption of the phrenic nerve. Next, in the lower half of the cervical spinal nerves is what's called the brachial plexus. And the brachial plexus has 5 different areas. There's the roots, the trunks, the divisions, the cords, and then the branches. And so the way you can remember that is, I don't know who the hell Robert Taylor is, but the guy who taught this to me in anatomy many years ago, he said, Robert Taylor drinks cold beer. I have no idea who that is, but I guess he drinks cold beer and he's happy about it. So roots, trunks, divisions, cords, branches. They all have names, which we're not even going to begin to think about now. Then there's the thoracic spinal nerves. Oh, I guess I don't have that cross-section. Okay. 12 of those, 12 thoracic vertebra, 12 nerves. Lumbar, 5 lumbar vertebra, 5 lumbar spinal nerves. And then 5 sacral and 1 coccygeal nerve as well. Okay. So all of that coalesces into what's called the sciatic nerve, the large nerve that serves the lower extremity. The sciatic nerve is important clinically for a couple reasons, and that's the reason I'm putting this slide up. I guess I should have a better picture to depict that, but you can get something called sciatica. I guess I've talked about that in lab already. An inflammation of the piriformis muscle, deep in the pelvis, which can press on the sciatic nerve and cause pain and weakness in the leg. Happens from sitting in chairs like you guys. The other problem with the sciatic nerve is when people are giving hypodermal injections in the butt. It used to be often that's a place where you're getting a big injection, a big fluid volume of an injection. You better know where you're going because you do not want to inject it right into the sciatic nerve. It's important to be able to locate the sciatic nerve because it's a not uncommon clinically imposed malady. So the doctor, what is it? What is the term? Iotropic? What's the term when a doctor causes the harm? There's a word for that. I'm blocking the word right now. Anyways, you want to make sure you know where it is and know how to locate it because it can be a common problem. All right. So what I really want to talk about is the motor homunculus. So to get there, I need to talk about a little bit of this business here. When you are thinking about the spinal cord, the posterior half of it is all sensory. So this dorsal root ganglion, these are going to be sensory neurons. All of this is sensory. There's going to be somatic and visceral. There's going to be visceral sensory neurons synapsing in the ganglion because somatic nerves don't synapse outside of the nervous system. They go directly to the muscle they're innervating. But visceral sensory neurons do synapse. There's a presynaptic and a postsynaptic neuron. For the sensory nuclei, that happens in these dorsal root ganglions. And then everything on the anterior side, these anterior and lateral horns, this is all motor nuclei, and that's coming out the ventral root. So this is all sensory, this is all motor. Some half and half somatic visceral, half and half somatic visceral. They come together and they sort themselves out. Dorsal root does not mean that it's only servicing things on the back. It means that it's all sensory. Ventral root doesn't mean it's only servicing things on the front of the body. It means that it's all motor. It's going to sort itself out as a spinal nerve, and then there's going to be fibers that go to the back, and there's going to be fibers that go to the front. All right? Does that make some sense? Okay. So those are the nuclei in the horns. I'm just putting my coat on. I'm sorry. In the white matter, there are these tracks that are ascending and descending. So it's white matter. They are columns, as I had indicated in the lab, because they are axons that are myelinated. And you can have ascending tracks or descending tracks. And here's a little map of them. Ascending tracks are going to bring sensory information up to the brain, and descending tracks are going to bring motor information down from the brain. Efferent. I gave you some of these words in lab. I talked about decussation. That's the going from one side of the nervous system to the other is decussation. And here are two other, so I guess these kind of anatomical terminologies might have been good on the first day, but you're getting them now. Something that is contralateral is a referential term. So this ear is contralateral to, I don't know, this eye or something like that. They're on opposite sides of the body. Ipsilateral just means on the same side of the body. All right? That's all those terms mean. Okay, we're getting super close to the most interesting thing of the day. And I just have to talk about these sensory pathways, and then we'll be there. So when you're thinking about ascending tracts or neurons that are afferent neurons that are going up, there's this ordinality to them. There's a first-order neuron. A first-order neuron is the neuron that is actually doing the sensation. All right, it's actually doing the sensation. And the cell body of that is going to live in the dorsal root ganglion. All right, or if it's a cranial nerve, in a cranial nerve ganglion someplace. But we're not going to worry about that. So there's some sensory nerve someplace. It's cell body. So like this, the feeling that you're getting on your fingertips, the nerve, the cell body of that nerve lives near somewhere back in your spinal cord, near your spinal cord. All right? The cell body is there, and it comes in to the posterior gray horn where there's going to be a synapse on a second-order neuron. A second-order neuron is going to, the cell body is in the posterior gray horn, and it's going to ascend. Usually it's going to decussate through one of the commissures here, one of the gray commissures. It's going to decussate and then ascend a tract, specifically the spinothalamic tract, meaning that it's going along the spine up to the thalamus. Now, what did I say about the thalamus? I said something about the thalamus.
All the sensory or like anything comes in and it relates back to the brain.
Yeah, if we're going to know that something happened, if we're going to feel a sensation, it's got to synapse in the thalamus. All right? So the spinothalamic tract gets us from the spine to the thalamus. This is the second-order neuron. It's going to synapse in the thalamus, and then it's going to project out to the cortex, all right, where we are going to then be aware of it. And that's the third-order neuron, the cell body of which is in the thalamus. All right, here they are. So these sensory pathways were first mapped out by this guy. For any of you interested in the brain or neuroscience at all, this is another delightful read. You've gotten two of them today. The Oliver Sacks book, The Man Who Mistook His Wife for His Hat, and about 3 or 4 other books that he wrote. And then this guy, Wilder Penfield, he is a really delightful character, Canadian, that studied at Oxford first he went to Princeton. This is where I guess he played rugby at Princeton. But he then went on to study with a guy named William Osler at Oxford, and then at New York with Harvey Cushing. For those of you who become physicians sometime in your future when you have more time, you should familiarize yourself with Osler and Cushing. They're both very famous physicians of the 20th century. They did a lot of foundational work. Anyways, he worked with the best. Smart guy, lovely person. Went on back to his native Canada to be at McGill. And while there, he wrote this book called Mysteries of the Mind, which is the book that I suggest you read. It's really fun. But in that, he talks about his experiments. And what this guy did was he would conduct brain surgery. He would do a calvarectomy. He would basically take the top off people's skulls while they were awake, and they'd have a tumor. He'd remove the tumor or whatever, and then he would take electrodes and he would poke and prod at the person's brain and ask them what they were experiencing while he was doing it. They would say, "Oh, that I feel my lip feels funny now," or their arm would move, or they would smell something weird. And in so doing, he was able to map out the sensory homunculus. So what you see here is his homunculus. And this is indeed a tiny little map of the body that lays on the surface of the brain. And the reason it has such strange proportions is that the amount of the brain that was devoted to a particular region was directly proportional to the neuronal density in that part of the body. So you see here the sensory pathways. You have a lot of neurons concentrated in the hand, the lips, all right, the face, the ears. Not so much in your back, for example. So you have a lot more of the brain dedicated to sensory mapping of the mouth, the tongue, the lips, the hands. All right. So let's see a little picture of it. It turns out that this part of the brain is this post-central gyrus. Here's the central sulcus. Here's the post-central gyrus. And they call this the PSC, the primary somatosensory cortex. This is where the homunculus resides. So the area of the primary sensory cortex is directly proportional to the density of the sensory receptors for that part of the body. This is an important concept. This is kind of like the only genuinely interesting thing I've talked about today. That's not true. I find this fascinating. All right. So we're going to next talk about the precentral gyrus. And everything so far has been about getting up to the brain. The magic happens in there. And then we're not going to talk about the magic so much. Then we just want to talk about how to send a signal to the effectors in the rest of the body. This is the motor homunculus. So the precentral gyrus is the primary motor cortex. And just like in the sensory homunculus, there is this representation of the effectors in the body, mostly muscles. And the specific amount of area in the primary motor cortex is directly proportional to the density of motor units in that part of the body. So if you look at it, we have a tremendous amount of area dedicated to the hands. We're gathering a lot of information in the hands, but we have a lot of fine motor control over the hands. Similar with the mouth, tongue, people use a lot. There's a lot of fine control that dictates speech, and of course, the larynx. All right, so all of that is here. There's a little sensory map of the body here and a motor map of the body there, the specific area of which corresponds to motor density. All right. So let's close the loop. We said when going up, there were first, second, and third order neurons. First order is the extrasensory neuron. Second neuron has its cell body in the posterior gray horn and decussates, ascends the lateral spinal thalamic tract up to the thalamus. And then you have a synapse in the thalamus and a projection to the cortex, specifically to the primary somatosensory cortex. Now, to go the other direction, it's going to take two neurons rather than three. There's going to be upper motor neuron and a lower motor neuron. An upper motor neuron and a lower motor neuron. These are general somatic efferent fibers, GSE fibers, that are going to, the upper motor neurons, both of them are GSE fibers, but the upper motor neuron is going to synapse. If it's a cranial nerve, it's going to synapse in the somatic motor nuclei in the brainstem, and if it is a spinal nerve, then it's going to synapse in the spinal cord, in the gray matter in the spinal cord. And then once it does that, then it projects out to a skeletal muscle versus via the lower motor neuron. So one of the things I want you to notice here is that if it's synapsing in the brainstem and coming out via cranial nerve, it does not decussate. It does not decussate. And if it's coming out through a spinal nerve, it does decussate. All right? That may seem like a super minor thing to you, but it turns out that if you know that fact and you know some basic neuroanatomy, when somebody comes in the clinic with who's had a stroke or has some kind of a lesion in their brain somewhere, depending upon the pattern of impairment, you can say with pretty good precision, without doing any imaging, where that lesion is. It's either going to be an upper motor neuron syndrome or a lower motor neuron syndrome. So maybe you have like face drop on this side, which is being controlled by a cranial nerve, but you have paralysis on this side, right? Or maybe all of your symptoms are on one side of the body, all right? So this is going to tell you where in the central nervous system your lesion is, the pattern of problems, all right? If it's an upper motor neuron syndrome, maybe you have a lesion over here or up in the PMC someplace. And so you're going to get this contralateral pattern in the face and in the other side of the body. Whereas if it's a lower motor neuron lesion someplace, or if it's below this level, it's down here, all your pathology is going to be on one side of the body. Does that make sense? All right. We talked about this in lab. I'm not going to talk about it again here because I only got a few minutes. This is pretty interesting. So as the motor neurons come down, they go through the various plexuses as they emerge through the spinal nerves, and they sort themselves out into the terminal nerve branches. As these reach the skin, we have this dermatomal map. This dermatomal map that is described by these sensory neurons, all right? So the thing that's interesting about dermatomal maps, so for example, you can see here L or T11, thoracic nerve 11, wraps around this part and up onto the back side of the body there. You have this sort of striped pattern across the body. What's interesting is the pathology associated with this. So shingles. Does anybody know anybody who's had shingles? Yeah, it's not fun. Certainly not fun. I've had it before. You get it from the varicella zoster that causes chickenpox. I had chickenpox when I was a kid. You guys probably got vaccinated against it. But the virus lives in the cell body of the sensory neuron, all right, in the dorsal root ganglion. And when you have an inflammation, the virus in that particular ganglion erupts and then causes an inflammation along all of the sensory nerves on that dermatomal map for that particular nerve root, all right? So when you see a rash that is on one side of the body and is only in a single dermatome, that's shingles. That's shingles. Kind of interesting. Referred pain. This is the interoceptors. Do I want to talk about that? I don't think I do. And the neuro. I only have a few minutes here. Went slower. Neurotransmitters. Let's not worry about that. Yeah, this is good stuff, but I don't want to rush through it. Classification of reflexes. So again, let's not sweat the details. Let's just talk about what a neural reflex even is. 10 more minutes, I would have been okay. 10 more minutes. So step one, we have some kind of a stimulus. So what I guess the first thing to say is, what is a reflex? Reflexes, you need some kind of stimulus to elicit a reflex. By their very nature, they're quick, they're involuntary, you do not have control over them, and they are stereotyped, meaning that whatever stimulus you're getting, there's a stereotyped response. There's a response that is not like specifically adapted to the situation at hand. It's just like that's what you get, automatic response. All right. So you get the stimulus. You have the first order neuron sends the signal in through the dorsal root to synapse in the posterior gray matter. You have the second order neuron, which is going to take the signal, and it's going to do two things. It's going to have the first part of it is going to synapse on the third order neuron. It's going to come right out and hit the effector that we need, all right, the muscle. But it's going to send a collateral branch, what's called a collateral branch. Another branch is going to decussate and ascend along the lateral spinal thalamic tract up to the thalamus so that we know that something happened. All right, I stepped on a tack there. I put my hand on something. And I don't want to like step on it and think, oh man, that hurts. I should take my foot off of it. I want to jump off of it right away, as quickly as possible. So a reflex is built so that you can get an automatic response right away to deal with an emergency situation. And also send information upstairs so that we can have voluntary input and whatever else needs to happen. All right? But right away, you're going to have an immediate response. Okay? Information processing happens in the CNS, activation of motor neuron. Okay, so we got all that. And then the response of the effector. We take our hand off. All right. So I'm not even going to go through any of these at all, unfortunately, because time is basically out. Babinski is a really interesting thing. But here's the recap. I went through the hierarchy of the nervous system, CNS, PNS, brain, spinal cord, and then PNS, somatics, and autonomics. I talked about the difference between afferent and efferent. I went through a few of the different types of afferent sensory receptors, and I talked about the four different types of information they convey: modality, location, intensity, and duration. And then I talked about the CNS, went through glial cells, and then the anatomy. Again, it was more anatomy than anything of the CNS. I talked about how the cerebral spinal fluid circulates. And then I talked a little bit about dermatomes. The important thing, I guess, I talked about were the sensory and motor pathways and the homunculus and the mapping of all that on the brain. And then I didn't actually get a chance to talk about any of that, really. I just gave you a very brief overview of what a reflex is. But that's a lecture, long enough anyways. I appreciate your attention and your earnest hard work. I hope you have a great day. Please get some rest. Go for a walk. Go to the gym. Take a swim. go skiing out of Quarry Road, although it's really icy. It's not ideal skiing conditions.